a motor of competitivity for our surrounding.
An opportunity to really study complex molecules.
My name is Montserrat Terrazas, and I am an associate professor in the organic chemistry section at the Faculty of Chemistry of the University of Barcelona, Spain, where I also lead the chemical biology and therapeutic applications of nucleic acids group, which is a young research group that was established 3 years ago.
My name is Miquel Pons. I am a professor in the University of Barcelona. I am the scientific director of the NMR facility and a group leader in biomolecular NMR.
I am Dr. Marga Gairi. I am NMR specialist at the NMR facility of the Scientific and Technological Centers of University of Barcelona.
That was a nice coincidence, and in fact our group had started a project independently on monoclonal antibodies, and then one day Marshall Piatto from Bruker contacted us and say, well, you know, would you be willing to collaborate with us with this monoclonal antibody project? I said, of course, this is a win-win situation.
Having high-sensitive probes like cryoprobes open us the opportunity to explore molecules that maybe before, without such powerful techniques, we couldn't address. And so we started to think about with Miquel Pons, and it was nice because then Bruker proposed us to work in a project like that.
So we are collaborating with Bruker. We synthesize a model oligonucleotide, a very small molecule that contains the chemical modifications that are commonly found in oligonucleotide drugs.
Then we do the measurements in the gigahertz. I think that was a very, very nice setup in which I think we are all benefiting. I mean, we are opening a new market. There are many companies around, so it's important for us to be able to provide them with the technology needed to be on the forefront. And so we identify that as one of these technologies.
So oligonucleotide molecules such as antisense oligonucleotides and short interfering RNAs have great therapeutic potential because they can inhibit the expression of virtually any protein in the human genome, thus allowing to reach proteins that are typically inaccessible despite the therapeutic potential of these oligonucleotide drugs. The clinical applications faces important problems because they are not stable in plasma. We need chemical modifications in order to improve their biological properties. There is a clear necessity for analytical methods to characterize these molecules.
We get a high-FF instrument because that has a direct impact on the competitivity of our environment. So biotechnology industry category is important, and I think we are contributing to that.
Well, the main goal is to be able to use high-field NMR, the 1 GHz, to individually assign the different diastereomers that are present in the mixture.
With NMR, also, I think that in very complex molecules, what you can do is that you can study these molecules in their natural environment. For example, with monoclonal antibodies, you can study the molecule under the formulation conditions. So I think that in this particular field, NMR has not, let's say, a rival.